Hey, good morning. It's great to be here. It's always good to be at ISMB. My academic career has actually started quite close to ISMB. It was ISMB 96. I think I gave one of my first talks. So it's been, and I've been coming to ISMB ever since. So it's been really great to come to ISMB and meet old friends and make some new ones and start some collaborations with folks. So, it's a real pleasure. So what I'm going to talk about is computational biology in medicine. All of you guys produce fantastic data and methods and tools. I'm going to show you another use case of it. I think Zach Kohane showed you what impact it could have in the healthcare system. I'm going to show you what impact it's already having in the pharmaceuticals area, right? How the data and tools you guys produce actually gets leveraged in some ways by us. And I'm going to hopefully convince you that the work you guys do extends beyond basic science and beyond the healthcare system, also into the pharmaceutical industry. So my story begins with this animal's picture who you see, which is a Norwegian lemming, right? And this is an animal that's reputed to march into the ocean in vast hordes when its population rises beyond a certain point. Basically say, look, this is not sustainable, and they just go into the ocean and drown themselves, or so it's reputed. And often it was said in the press and other places that, the drug industry, there are only a few targets in there and everyone's basically going after the same targets and there's really not much novelty in there. So we started looking at this problem a little while ago, and try as we might, we couldn't really find references to where this was actually coming from. What was actually the data around it? So we dug into this data a little bit, and for this we use this commercial source of industry projects, which is probably a little bit more comprehensive than some of the public sources out there. This is from a company called Pharma. It's called Pharma Projects. And at the time we looked at it, it had projects. It has over about 65,000 total projects. This is any drug project that's ever been conceived of or publicly disclosed by any entity, whether it's commercial, in the public domain, universities, funding agencies, anything else. And when we looked at this data, it had about 251 or around 1,000 human targets that were the subject of the active compounds and drugs, right? And about only of these 251 targets, we focused our attention on the ones that were reputed to be selective, right? That the drug was only hitting a single protein, or at least in this database was only a single protein and annotated as the target of that drug. And those were about 1,000, and that's what we chose to focus our attention on. It's not because the others are not important, it's just that the data is a little bit cleaner and it's probably easier to draw inferences from it. And we split this set into two categories. One, what we call proven targets. Those are targets for approved drugs that are selective, right? So as I said, the database had to list that as a single target, but we also looked at novel targets, right? So these are the ones which are being targeted by drugs in the pipeline, so either in human testing or in preclinical pipelines. So those are the two categories, proven and novel. We left out a few other targets where, it wasn't quite clear how to put them into either of these two categories. But I think we will see that hopefully doesn't make a difference to the story. So the first thing we looked at was, when you look at these 1,027 targets on the left, right, what you can see is if you break it down by how many companies are looking at it or how many entities are looking at it. So remember, we looked at academics groups, we looked at commercial groups, biotechs, pharma. So it's basically how much competition is there in this space, right? So if you look at this, what we found was 42% of this huge blue sector, there was only one project or one company that was looking at it, right? So there's a fair bit of novelty just from there. You can see that it's not like everything is being worked on by everyone, right? And 1,000 targets, I don't know whether that number is large or small. It could certainly be larger, but it still represents a fair fraction of the human genome, right? And 26%, however, was still being worked on by 5 or more entities. And I suspect when people talk about, that everyone's looking after the same targets, those are those 26% are what people are actually talking about. And in fact, if you break this data down by the proven and novel targets, right, you can see that the proven targets, the 247 of them, that's where really all this overlap is, or you see these are the targets of marketed drugs. And you can see once there is a marketed drug against a target, it makes sense for other people to look at it and see if they can find a better modality, a better treatment, a better compound that can provide increased patient benefit. So you can see that there would be a lot more competition around that. That target, I mean, is ultimately validated, right? If you have a marketed drug against it, you're not going to get better target validation than that. But even there are some small fraction of those that, those are a little bit more esoteric and perhaps not that many companies are looking at it. But this is the part that I think is the most interesting on this figure, is the 712 novel targets. So these are targets that no marketed drug exists against that target yet. And 54% of them, a majority of them actually have just a single biotech or company looking at it. So what that really says, people are trying to explore new areas of biology and trying to look at these targets and see if we can relate them to disease and can you find compounds against them that can be used to treat them. And but there is even there are a few that actually are fairly well worked over. The other thing you can do with this kind of data is you can break it down by phase. So if you look at these projects or targets by phase, you can see in preclinical, for example, there are 229 targets that are worked on by one or more company. And as you advance in phase, as you go into phase 1, which is mostly safety, phase 2, where you look at a small signal in terms of efficacy, and phase 3, where you really do these large, well-powered statistical studies to show clinical benefit, you can see that the number of novel targets really decreases. So what that to us says is that, as people gather more evidence to advance the compound into the clinic, this is all publicly disclosed findings, et cetera. So other companies, other teams do start looking at those targets. So as you get more validation, you're going to get more competition, right? I mean, and that makes good sense. So it's not that all the groups just tend to focus on a few things. It's just that as evidence builds up, so does people's interest in that target and exploring the biology around it. The other thing I want to point out on this slide is that the number of targets that are actually in these phases is actually quite interesting. So there are 83 new targets in phase 3. So that means that there are 83 sort of novel mechanisms of action, at least 83 novel mechanisms of action drugs being targeted that could actually find their way into the hands of MDs and patients, hopefully relatively soon. And there are 214 such targets even in phase 2. So there is a fair bit of novelty in the pipeline, right? In the pipeline, this is across the whole. This is across the whole world, the entire industry, the entire academic world, the entire. It's basically everything you got, right? So, but this does not answer the question as to how do you validate these targets? How do you find new targets, right? Often, I work at a pharmaceutical company, and teams will come to me and say, "Can you give us a novel validated target?" And that's an oxymoron. Either a target is going to be validated or it's going to be novel. If it's validated, people will know about it. I mean, how can I make sure that this is only private to you? Everyone else knows about it. Because our sources of information, as I say, are the same for the most part as in the public domain as the data you guys produce, analyze, and put out there, right? So that's the primary source of information. But at the same time, what GSK is doing is we have a partnership with EBI and Sanger called the Center for Therapeutic Target Validation, where pre-competitively we are exploring how can we get better target validation. Ewan Burney is the interim head of it, and in fact, they're looking for a full-time head, so if somebody's interested, I think Dominic might be willing to take names. And they also have a website where you can start exploring what the information around target validation is, how do we do target validation, and hopefully this community builds up, and in the public domain, there is enough information around target validation that it can really help drug discovery and development efforts. But there's a special kind of target validation that I'm particularly interested in, and which I guess the field calls drug repositioning. So computationally, as computer scientists and as informaticians, to us, it really does not make that much of a difference, whether you call it target validation or repositioning, because it's essentially the same problem. Given a human protein, what diseases is it associated with? So if we modulate it using some small molecule or antibody, which patients will benefit, right? So it's the same question, except when you're doing drug repositioning against this human protein, you already happen to have a good molecular entity, right? So it's a much more restricted problem, but the advantage is that you have more proximal value. So if you validate a target now, say you came up with some beautiful, nice scheme to validate a target now, it'll still take 10 years, 15 years to actually show that what you did actually results in some clinical benefit. Repositioning can hopefully shorten that time down, because if there's already a clinical asset, you might be able to get there quicker. So that's the key point here. And the data for repurposing, you can imagine using all kinds of data for repurposing, most of it in the primary literature, and it's the data that you guys produce. It's using the methods you guys produce, right? So this is just a big thank you from us and everyone else that, keep producing the data, keep analyzing it, keep finding ways, cleaning, curating it up, because there is utility for it, right? Anytime you take a protein and you find information around it as to how it relates to a disease, how it does not relate to a disease, it adds value. So, and we utilize data arising from all phases of the pipeline, arising from all kinds of technologies that you guys are involved with, from molecular assays to phenotypic assays, in vivo models, clinical trials, prescriptions. So there is a whole range of data out there that gets produced and analyzed, and I'll show you some examples of how we can use it. So the great thing is it's all in the scientific literature, so it can be extracted. A lot of it you guys curate and clean up and put into databases, which is always fantastic. When you add standard ontologies and definitions, again, that works really well because it makes it so much easier for us to use it. So the first method I'm going to talk about a little is that we have been using is the connectivity map. In fact, it came from this area around 8 years ago, 2006. This is a science paper from Justin Lamb, Todd Golub, Eric Lander, and others. And I'm going to use this schematic to show some of these methods that we're using. And it's essentially a very simplified view of a drug related to a target, which relates to a phenotype and relates to a disease. So what the connectivity map does is it provides a way of looking at a drug and measuring a whole set of phenotypes on it, mostly gene expression changes. So you dose a cell line with that drug and you measure which genes go up and which genes go down. And then the idea is that you can relate it to a disease, right? So diseases also have genes going up and down, and you can think of barcoding your disease and barcoding your drug, and then comparing these two barcodes and deciding which disease would benefit most from that drug. So what we have always liked about this, it's a holistic technique. It does not assume that you know all the biology, right? Drugs are doing things that we don't necessarily always understand. Diseases involve factors and characteristics we may not always understand, but as long as you can find a way to match them, and this just happens to be one of them, it might provide power, it might provide some utility, and maybe it provides some hypotheses that can be tracked down. So what we have always been trying to do with connectivity map is to try and understand how well it works, right? It's okay, you can always use the technique. I think there are over 1,000 citations to connectivity map at this point, or people using connectivity map to find various hypotheses and either doing some experiments to test them or at least speculating and finding literature support for their hypothesis. But it's really hard to figure out, does it work in a robust, systematic way? So what we have been trying to do, and I'm not sure we have been completely successful, is build a standard data set, right? Given a set of drugs with standard disease indications and standard disease signatures, how well can you do this matching, right? So this is something that's not published yet, but, and what we have found that using some a metric that we call extreme sum or X sum, where we essentially some of the fold changes on the most extreme genes, we find that you can get some signal in terms of doing this in a robust, systematic way. I mean, this is certainly not going with the best technique out there, and it's probably not the most, but the key thing we are looking for here, right, for something to be practical, the area under the curve for the ROC is not useful, right? You want very high precision for the early part of your retrieval, right? So if you predict, give me 10 hypotheses and tell me 2 are true, right? That's actionable, because then we can go and test those out. But if you give me 1,000,000 hypotheses and say 10% are true, I mean, that's not, or even 90% are true, that's not very useful, because I have to test 1,000,000 to get and that's not practical. So so when you develop these methods to say, look, this target is associated to this disease, please focus on precision in the early part of your curves, right? Because that really makes it practical, makes something testable. And then we can go and act on it and see if those things hold up. So the second thing I'm going to discuss is genome-wide association studies. And again, what the genome-wide association studies do is they connect a target to a phenotype using a polymorphism. right? And the tricky part here is that which gene is connected to the SNP is not quite clear, right? And this seems to keep changing with every technology. In fact, just in the last 2 days, I've heard at least 3 talks which sort of changed my view on what would be a nice way of connecting the SNP to a gene. So you could use the nearest gene, which is what the NHGRI GWAS catalog does. It's mostly the reported gene by the publication themselves. You could use data from things like ENCODE and Phantom5. You could use EQTLs. And there were some other talks yesterday that showed even perhaps better ways of connecting that. But again, this is the kind of data that we need. If you can form connections between these polymorphisms and genes, and that then we happen to have a drug that addresses that gene or downregulates that gene or upregulates that gene or binds directly to that gene, then you have a testable hypothesis. You can say, look, does that drug affect that disease? And that's something we would love to test and find out. And human genetics is one of the best sources of target validation out there. I think a lot of pharma biotechs might disagree on what we believe and what we don't believe in terms of target validation. The one thing you'll find common consensus on is human genetics, right? Things that relate GWAS SNPs to the genes and all the progress that's made therein is extremely useful and critical. And in fact, we formalized this thing a few years ago where we looked at all published GWAS studies, and we essentially did this really complicated computational technique called set intersection to see which genes had phenotypes and which genes also were part of a drug target to see what the overlap was, and said, look, is there something good we can get out of that? And we found there were 63 genes where the indication for the drug matched what the GWAS was predicting or close to predicting. And there were 92 other instances where, it gave us a new idea as to what other indications that could be used for. And this is clearly supported by human genetics, provided that SNP is actually correctly related to the right gene, right? So the third thing I'm going to briefly discuss is how do you use screening data? Now, there's a lot of screening data generated both in the public domain and things like PubChem, as well as in the industry, where you take targets and you screen them against a whole library of molecules, and you find which compounds are active, which compounds are not. Also, you do a lot of phenotypic screens, right? So you might find that there are compounds that are active against the phenotype or the target, but then there are instances where you can also find the same compounds that are active against a target or are active against the phenotype, right? There is overlap between these two, right? And often that can give you a new hypothesis that, look, if you happen to have a molecule against that target, a clinical compound might already exist. You can say, look, this clinical compound binds to this target, and you might be able to affect the course of this phenotype or that's related to that disease through this connection. And in fact, this is an interesting story because I gave a talk at ISMB in 2010, and I really need to give that acknowledgement. And after my talk, Josh Swamidas, who's the first author on this, he came up to me and said, this is very interesting, maybe we can work together. And this collaboration came as a result of that talk. So for people who hesitate to, talk, if you have ideas as to how you can work together with us or with other speakers, it's really worth, we come to the meetings to talk to people and not just listen to presentations, because you could just hear them on video or audio, right? You can collect all the hypotheses you want, but they might be nice papers, but that's good value maybe, but it's limited value. What we try to do is, once we find these hypotheses, is we really use a variety of techniques to gather evidence. Thanks. A variety of techniques to gather evidence for them, and we use internal search engines. Lots of times you find that people have already tested parts of that hypothesis internally, right? They might have already done that assay that says this compound does reduce TNF alpha, right? So you can find internal data, you can find public data. Many of our compounds and other companies' compounds people are testing and doing things with them all the time, and you can find lots of evidence out there to support or to say, look, this hypothesis doesn't really work. That's already out there, and that's always the best kind of thing, because if you can gather all that data, figure out what's going to work, what's not going to work, you can place your bets, right? And we really believe in pharmacological data in that space, because genetics just tells you that that at birth, if you happen to have one allele versus another, your chance of getting a disease might be high or low depending on what it is. It doesn't tell you if you pharmacologically intervene, you will be able to affect the course of that disease, right? So pharmacological evidence is really very compelling, in addition to when coupled with genetic evidence. And typically what we do is we'll work with internal groups to either get them to test it, or we work with in external partnerships to get other people out there to test it. And sometimes we just test it ourselves. So, computational biology, and I see Atul back there, he's been the pioneer in this regard about, we can own this space. There's no reason why we can't do the testing ourselves. So if you have good ideas, you come up with a hypothesis, you can't convince somebody else to test it, go ahead and just test it yourself. And I think that days definitely come. But the stories are not always that simple. What we want to do is get drugs to patients, right? We are not just about getting a hypothesis that looks nice. And most of these ideas don't work, right? So I'm not going to lie to you and say every hypothesis that we come up with is brilliant and will work, right? Most of them we are able to discard even before we test it because you find serious flaws with it, right? Just because something works, something sounds good in theory doesn't mean all the things line up. And most of the time, there'll always be a critical flaw that makes it not a good idea. And that's one of the things that is fairly hard to do unless you have some experience and knowledge around what is the drug, what are its activities, what do we know about it, what is the target, the disease population, can you actually get it into the right tissue, or what's the PK? I mean, there are a lot of questions that come in. And there's an excellent book on drug repositioning, actually, that I'll encourage people to go read up if they're interested in this. And of course, it all comes down to talking to people who know about this, right? So we certainly don't know about it. I'm a computational biologist like you, and probably know just a little bit less or more than you do on most of these issues, right? And of course, IP is another important matter. For a marketed drug, if you find a new indication, it's typically hard to make that work and for somebody to spend all that money to develop a new indication. But the advantage of working in a pharmaceutical company is that you actually have IP protected pipeline. So if you have something in clinical testing, you find another indication for it, there is a path to the clinic that's much easier. So that really makes it a much better proposition. And at the end of it, we are constrained by our own time. So of all the hypotheses we come up with, the advantage or disadvantage of computational work is, you crank the wheel and out pops so many hypotheses. How do you pick among them, right? So, you have to be able to prioritize among those. So my takeaways, if you will, are, I think systematic repurposing can work. In fact, we have some evidence to show that it can work. It requires persistence and resilience. It's not easy, and it requires us as computational biologists to get out of our comfort zone and learn about a lot more things than we are quite comfortable with, right? You guys produce fantastic data, so, but the more you can clean it, curate it, ontologically label it, annotate it, the easier it makes it for us to use it, leverage it, and show value from what you're doing. And the other thing that we see is simple methods that are intuitive and reproducible are, in my opinion, always better and easier for us to use than there is a trend in publications that, there's 6 different complex procedures applied, and it's very hard, even as a reviewer, to understand what the value of any of those is, right? And I think that's something we need to, maybe the field needs to back away a little from and say, look, if the method is more complex, there really needs to be stronger justification as to why a simpler method didn't really do the job, right? We welcome collaborations, people who want to work with us, and we are also looking to hire people. So, and we'll stop here and take questions. Thank you.
Actually, 2 questions. You said that the most important is precision for the early part. What do you consider high precision there? You said 90% is not enough?
Oh, 90% was fantastic. Even 20% is high enough, right? So if you give me 5 hypotheses saying, look, if you test these 5 compounds, one of them will work, that's a good bet because.
Okay, so I thought you said that.
So no, but that's a great question to ask because in drug discovery, most projects fail. So you're not battling against. In fact, I automatically reject any paper that says they can reposition with 90% precision, right? Because it's not possible. Right? It's just that would be pretty fantastic, right? Right. So.
Now, a second question is, I think most of what you're discussing has to do with small molecules. What about repositioning of antibodies and other biologics? Is it the same logic, or is it a completely different story?
It's exactly the same logic. With things like maybe connectivity map, there isn't as validated a process for profiling antibodies, but with all the other technologies, I think it's pretty much the same logic. It's just It works. So the nice thing about doing it computationally is, once you have the wheel, you can crank it in so many different ways, and it's essentially the same wheel with maybe minor gear changes, right?
Repositioning sounds almost like the same problem as detecting unwanted side effects. Is this something that you're investigating as well in parallel?
So you are absolutely right. Especially with technologies like genetics, when you get an idea that this target is related to this disease, you don't know whether an antagonist or an agonist will make the disease better. And if you happen to have the wrong direction, then it could easily make it worse. So again, exactly the same problem. So the way I think about drug discovery, right, it's maybe 2 or 3 very simple steps. There's finding a clean, safe molecule. There's no such thing as a completely safe molecule, but there are safe molecules, right? And then there's a part of taking that molecule, figuring out what disease it can work with, right? And whether what disease is going to help, what diseases it might hurt. And then it's actually then giving it to those patients and seeing if you can observe the benefit. So, computationally, there are only a few problems out there, right? If you really want to think about it that way.
For single drug repositioning, I mean, why don't you just use cell lines to screen the drugs directly?
So if you had fantastic phenotypic assays, right?
Yeah.
So if there was a phenotypic assay that said in this cell line, if you observe this, that's a good readout for Alzheimer's disease. We would have cures for Alzheimer's disease within a few years. The problem is those assays don't exist. So, I mean, this is probably not for this audience, but coming up with a phenotypic assay or some kind of thing that tells you this drug's going to work, and if you have an experimental system or a computational system, I think we are game for either, but they don't exist.
Okay, but for most of cancer, we have a lot of cancer lines already, right? I mean,
so, and that's why there's a lot of effort around cancer, and you've seen so much progress around cancer is because of the cell line and the possible predictability of the cell lines to humans. But even there are some challenges which are probably too technical to go into.
thank you.
So you mentioned that most drugs fail. Is it because their efficacy problem? And if it's efficacy problem, is it because they're predicted based on their ability to target single protein rather than a function? And because we have available data like CMAP, maybe we can predict drugs based on their ability to target several disease-related cellular functions.
So drugs. So when we say most drugs fail, it's actually from the very early part of the process to the end, right? So some of them never even see. It's just hard to find drugs for some. You might screen 1 million compounds, but you don't necessarily get that many good hits, right? And with the hits you get, there are all kinds of reasons for failure. Efficacy is one of them. Safety is another. There are PK or other issues as well. So there's a whole bunch of reasons out there. But the one we are focused on is efficacy, right? Because that's where repurposing and target validation can help. And if you can figure out what the right target is maybe it helps. And what I hear you saying is maybe there is no such thing as a right target. It's a right mechanism or a right pathway or a right conglomeration of targets. And that's going to be some of the challenges for this field. I mean, that's something we need to think about. How do we deal with that? And how are you going to find a drug that does 70% of this and 30% of this and 40% of that? I mean, finding that drug is a little bit of a tricky problem, right? But I think you're essentially right.
Thank you.